Hello, everybody. Welcome back to our event. This is the final part of our four-part series introducing AI for image analysis, how to be successful. So indeed, we've been together for 4 days with Philipp Kainz and Birgit Müller. And I'm very happy today to be with you again to conclude this series. So for those of you who are joining us for the first time today, just a quick tour in Zoom. So all you need to be aware of is that you will find at the bottom of your screen the FAQ box. So currently you are all muted because we are in a Zoom webinar. However, you can at any time post a question into the box or a comment, and we will make time at the end of the session to answer those. So feel free to use this function throughout the session, and then we'll get back to you towards the end. So my name is Anne-France Carter. I work at Precipoint in the marketing team, and like I said, we've had a bit of a webinar marathon this week, and now we're reaching our final stage. So I've spent the last 4 days with Philipp Kainz and Birgit Müller. Philipp Kainz is founder and CEO of KML Vision. It's been a very interesting week as Philipp has been sharing his expertise on how to start an AI project. And very specifically, when it comes to choosing a software tool that supports your research needs. I'm also introducing my colleague, Birgit Müller, once again. Welcome, Birgit. Birgit is Head of Business Development at Precipoint. And I have also the great pleasure today to have Dr. David Hercher with us from the Ludwig Boltzmann Institute in Vienna. David is the head of Neuroregeneration group, and his main research focus is the investigation of regenerative processes after injuries, with a special focus on the nervous system. So you will hear more about it in just a minute, as David will be our main speaker today. After his presentation, we will have our usual Q&A session. So with this, David, the stage is yours, and I wish you all a very pleasant time with us today.
Thank you very much, Anne-France. It's a great pleasure to be here, actually. Anne-France, I think I have to share my screen.
Yes, please.
Perfect. So, my name is David Hercher. Great to be here. So a short backstory of me. So I studied molecular biology in Vienna and then did my doctorate at the Medical University in Vienna also. And currently I'm leading the Neuroregeneration group at the Ludwig Boltzmann Institute for Experimental and Clinical Traumatology. Quite a bulky name. So that's why I said it's the LBI for traumatology. Yeah. So where are we? We are in the hospital situated here in Vienna. It's a trauma hospital, and our main research focus is everything that happens after trauma or / infection. So we have several groups. Some of them tackle acute care, intensive care, but also novel surgery approaches. And finally, we try to follow the patients until they're fully healed, so follow up on regenerative processes. And my focus is the nervous system, with a special focus on the peripheral nervous system. Yes. So our motivation, and why am I here? Our motivation is a situation like that. Yeah. So trauma happens quite often in rural areas as well. Their saw injuries are quite often. Here we see an injured severed nerve, and what we want to achieve actually is something on the right-hand side, so full functionality. And there are several approaches to reach this goal. One of them is the tissue engineering approach, which is one of our approaches as well. Here, we try to restore the continuity of a severed nerve. This would be the nerve here in yellow, with tissue engineered conduits that could be of various matrices that can be filled with cells or with neurotrophic factors, in boosting the regeneration. But one big issue in the whole field is how to evaluate axonal regeneration precisely. There are a couple of approaches for this. So the old school ones would be histomorphometry, where you use osmium tetroxide in order to stain. Here you see axons, the myelin sheath around the axons. However, sorry, there's a fly. However, this is quite cumbersome from the fixation point of view. It only shows axons at later stages of regeneration and only myelinated fibers. So a couple of fibers, for example, all the pain fibers, C fibers, are not myelinated, so you miss out on them. Another approach is immunofluorescence. There you stain only, for example, axons. However, they have the drawback that they have shorter shelf life, and also the overall structure of the nerve is not visible. And it is not possible to stain, for example, for collagen as well and stuff like that. That's why we decided to stain our samples in our regeneration models with neurofilament and do normal immunohistochemistry. Here you see a cross-section of a nerve stained for neurofilament. So, but as you can imagine, the quantification of this, the manual counting of this is very cumbersome. So we reached out to KML Vision also, to Philip, and developed together a novel AI, which is capable of automatically identify axons in sections like these. So why artificial intelligence? As I said, the manual counting is very cumbersome. We have hundreds of slides. We have several thousand axons per slide. So you can imagine this is not really feasible. There are some semi-automated solutions available, like AxonJ, which is a plugin for ImageJ or PG. However, this is not really satisfactory. We tried everything, but it's not really satisfactory, the outcome of these. And there are no automated solutions available for these kind of immunohistochemical slides. So what does the workflow look like? Yeah, so we have preclinical research, for example, rat models, where we try new therapeutic approaches for nerve regeneration. We harvest the nerves with the conduits, for example, slice the nerves for histology, prepare them for histology, scan them, and digitalize them. And finally, we want to get some data out of that, quantitative data, and not only qualitative data, because that is mostly done so far. So how do we get quantitative data? And for this, I want to go more into detail from the stage when it's already digitalized and how we are going to use this, this ECOSA software from KML. So for this, I want to give you a short live video on how to identify these and how this looks like. So this is the ECOSA platform. It's all online. You can upload your images into, for example, a project, and you will have your image library with all your images in there. And I prepared some of these. For example, you can look at them online, which is nice because data storage is always an issue and you can't. So one of these pictures has, I don't know, 50 to 60 megabytes or even more. So and you don't want to carry around a 1 terabyte hard drive all the time and then drop it. It's just not an issue, just an issue. So as you can see, this would be the digitalized histological slide. where I implemented already some region of interests. If I zoom in here, you can see this would be a proximal part of the nerve. You can see here in brownish the single axons, and also this would be a part in this conduit. In this case, it would be a silk fibrin conduit, which we are investigating, whether it can promote axonal regeneration. And there you can see also these axons. So how to quantify these? So we are pretty straightforward. We can just go to image analysis, right on the right corner here, and analyze, for example, only the ROIs, so the region of interest. Otherwise, it would scan like the whole slide, and it would take quite a lot more time to actually finish the quantification. So you define only ROI analysis, and I prepared this as well. This you can look up here in the image analysis. So this is, for example, the finished job. Different jobs here. We can show the details of these analysis. And for example, we can look at which ROIs were labeled how. So for example, here's the ROI 1, ROI 2, ROI 3. However, we want quantitative data and not only nice ROIs. So what it gives you, this program, is a CSV file, sorry, which you can download. You can download the whole bulk of data here. And this looks like that, where you can see the results visualization, which I want to show you also here. So this would look like this, for example. And here you can see already all the recognized axons in the slide. And this is also enumerated. So every axon which is recognized has a specific number. This becomes important later on. And there you see, for example, the part in the silk fibrin looks like this. The quantification works quite well, and the recognition with a very high fidelity. So for example, if you look at this would be axons. This is not an axon for the human eye. This is already for untrained users. This would be already difficult to differentiate, something like that. However, this works nicely. And if I also get the CSV file, which I can import into Excel, where I will get data for each object. So I will get my ROIs, ROI 1 to ROI 3 in this case, all my objects. I will get as a result area in pixels. So you need to know your resolution, obviously. The perimeter, elongation, and a couple of other parameters which are important to identify. For example, elongated structures would be like axons cut, not like totally in cross section. So this gives you an idea of the orientation of the axon in the construct. But what is most important, obviously, is the total number of axons. And this is something here. So this is, for example, one silk fibrin conduit. ROI 1 would be the proximal part, so the healthy part of the nerve. ROI 2 would be the beginning of the conduit, and ROI 3 here would be on the latest stage of the conduit, so later, more distally. Yeah. What does this tell you? Switch this. Yeah. It tells you that the recognition of axons with this software we developed is really high. So you can see here the raw input, you see a prediction, you see the results, and here in red you see some false negatives, and in blue the false positives, although these are mostly due to not correct annotation of our side, yeah, so because we had to annotate these by hand. This would be a normal outcome file you will get. You can actually evaluate the circularity. So the closer to one this becomes, the closer to a perfect circle the structure is. And axons should be circular in structure in general. It defines also the elongation, and you can define the diameter of the axons with these parameters you get there, which gives you an idea of what kind of axon it is, what kind of fiber it is. Is it a large motor fiber, or is it a small C fiber, for example? So I did this very quickly, just to show you also some data, what we did. So I think this took me, I don't know, all in all, maybe 20 minutes or something, with the 3 pictures. So we had 3 nerves, actually, 3 different treatment paradigms. We had a muscle vein conduit, a silk conduit, and an autologous nerve transplant, which we want to compare and want to see if the regeneration works better in one or in the other. And what you can see here, this would be the proximal part, this would be the early graft part, this would be the late graft cross sections, and distal part. And if you look, for example, at silk here and here, it's not really logical, but actually it makes sense that you have a lower axon count before the conduit than in the conduit. But that makes sense because axons, when they regenerate, they sprout. And so one axon becomes a couple of axons. So if you only go for the total axon number, it will give you a wrong feeling of what is happening. So if you look only at these 2 graphs here, or bars, it would seem as if the silk would be a perfect conduit. However, if you look at later sections, you see that there are no axons anymore, right? So this gives you a very good idea of the regenerative process. Another outcome you will get by this is you can count unmyelinated axons, which is definitely beneficial or plus compared to the histomorphometry, where you see only the myelinated axons. And this allows you also to see earlier stages of regeneration, because axons are getting myelinated only later on in the regenerative process. So if you have early time points, this would maybe be a suitable method for you. You can, with the circularity and the ferrite diameter I mentioned before, you can actually produce such a size distribution graph where you see, okay, how large are these fibers? This also gives you an indication of how well regenerated they are and which kind of fibers are regenerating better than others, maybe. And it gives you also an idea of the orientation in your nerve conduit. I also wanted to give you a short list of considerations, because it was a back and forth with the training for this algorithm. Most importantly, I think Philip mentioned it already on Monday, and I guess also on the other days, the quality of the digitalized slide is most crucial, right? So shit in, shit out, right? So it's always like that. What we did is also the 4 eyes principle. So for annotations, for novel annotations, if you want to train a new algorithm, for example, it's not sufficient to have only one person doing it. And especially, for example, often students are set upon this task to do stuff like that, right? But maybe they are not as experienced. So always the 4 eyes principle, but I think that is self-explanatory. What I really can recommend is the freehand tool, which is available, which makes life easier, definitely, because with the mouse or even only with the touchpad, forget the touchpad. Yeah, you will get. It's just not really. It's very cumbersome. But the freehand tool works perfectly, and I would definitely mention the ROI. So you can also only analyze specific ROIs of your slices. You can say, okay, I want to see only the axons inside of the conduit, nothing which is going on the periphery or for any other application. So my conclusion would be that I'm very satisfied. We have a very high fidelity of recognition and accuracy of the program so far. The bulk processing is possible, so you don't have to do it like I did when I showed it to you, so to analyze every slide after the other, but you can like analyze all your slides at once. Yeah, you just have to set the Royce and it's done. So it allows really fast quantification of axons, which are stained for this neurofilament with the neurofilament antibody. And it, therefore, as it is automated, it allows a really high reproducibility. And also what we really like about it, and why we also used these kind of older technique with the immunohistochemistry, was because we have a bunch of older experiments and slides from older experiments lying around, and which have been only quantified or qualitatively evaluated so far. So we can go back actually and quantify this as soon as we have digitalized them. Yes, and with this, I would like to thank my team actually, and Philip and everyone else from KML Vision and PressePoint for this webinar. Yes, and I'm happy to take any questions.
All right, thank you so much, David. It blows my mind every time I hear about artificial intelligence. Thank you so much. I think we do have questions. Yes?
Yes, we do.
So let's start, Birgit.
Exactly, let's do that. Thank you, David, from my side as well. I always think, you know, when you talk about it in theory, it already sounds good, but when you see then how you actually work with it on a real case, right, all of a sudden it becomes so much more clear what it is that you're doing and why. So let's look into a few questions that we have. I think there's one for Philip. The question is, which AI architecture did you use for this case?
So we tried a couple of them. Usually we have our own model that really works well on a huge variety of use cases. So we tried to, over the last couple of years, standardize all these pipelines. So we have a proprietary architecture. I'm not able to share with you in detail, but it's basically a fully convolutional neural network with some special tweaks to make it work on really small data sets as well.
Interesting. Then there's one question around whether you have, also for Philip again, some applications that can be used in routine pathology or in pathology generally, also, for example, with cytology.
Yeah, we don't have it right now developed, so there's a possibility to develop custom solutions. We don't have these off the shelf right now, but as you see that probably in a couple of weeks and some small iterations, you will be able to achieve an AI solution also for your use case. But it depends on where you want to use it. Right now, we don't have a certification for use in clinical settings, but that is something we aspire in the future.
Then I have a question to David, and that was, how did the process generally work with you in the interaction with Philip to figure out what is it that you need? Did you basically already know, okay, I need exactly this analysis, and then go to Philip and say, please do? Or was that more of an iterative process?
So the first step was obviously we had a problem with, we didn't want to use up half a year of a master student's time counting axons, but we obviously knew exactly what we wanted to have. We wanted to have something which is counting our axons and reliably recognizing them, preferably in a digitalized manner, yeah, obviously. And then, yeah, we came together, and the process was like this, that Philip and Michael, they gave us more or less regions of interest in our slides. We provided them with our raw data, in this case, the slides, digitalized, and they defined specific regions of interest on this slide, which we had to annotate the axons. So, for example, also a white background. So obviously there's no axon there, but also trickier sections of the slides where it was not really clear for an unexperienced person to identify the axons or differentiate the axons from other structures. So, and then it was a back and forth, more or less, and we had a couple of iterations and also feedbacks of the handling of the program. And yes, it was quite straightforward, actually. I don't know, Philip, if you want to add something.
No, I think you pretty much explained it. This is the process I also was referring to in my last talk. So there is a learning on both sides. Even, I mean, with every new use case we experience in the field, we need to be ready to be ready to learn on both sides. And sometimes it makes sense that we jointly define, for example, like in this case, the regions of interest that should be annotated, because we have the experience, for example, to select regions that are not too similar in order to maximize the output for the AI training in the end. So otherwise, the model would just learn always the same and has no chance to learn a general representation of the input.
Follow-up question to that, Philip. How did you augment the data?
So we had some staining variation, which we could measure from the data David provided to us. So actually, we took a couple of samples from the slides, made some color statistics, for example, and defined these boundaries that I was talking about, in which space you, for example, can augment your data. What is realistic and what can you expect? So we had some global and some local spatial and color transformations applied to the input in order to increase the data quantity for training.
Maybe very interesting, cool approach, right? Then I have one question that's actually coming from me personally. David, now that you've worked with AI, would you go back again and do it without it?
In this case, not at all, no. No, but obviously it really depends on your question, right? So for some research questions, qualitative analysis is really more than enough. And sometimes always pushing the numbers is maybe misleading. Yeah? Sometimes a good picture shows you more. Yeah. But for these specific questions, I wouldn't go back. Yeah? And I also have a couple of other research questions and in my mind, and we're also working on these to define maybe novel artificial intelligence solutions for these histological evaluation. So I think especially in the histological evaluation, it saves so much time. Yeah. But you have obviously you always have to have in mind that it may be a flawed, or not flawed, but it may make mistakes. Yeah. So always double check. Don't believe everything, but I mean, you wouldn't do that if your student would count that as well. So yeah, I think that's always important to have in mind. Yeah. The computer can only do or the AI can only do what we showed them in the beginning, as Philip said. So if I train them on the wrong set of slides and then just go ahead and put in 5,000 other slides, I cannot expect to have a great result. Yeah.
Absolutely. I think that was it in terms of the questions.
Yes, that was it. I can't see anything else. So yeah, we are actually really on time today. So just from my side, first I would like to really thank everybody on the panel for spending the week with us or the day with us. And of course our listeners, thank you very much. I hope you really enjoyed it. Feel free to reach out to us. Or stay in contact with us if you want to hear more about our next events that are coming up. You may subscribe to our newsletter or just check our webpage and look at the webinars that are advertised on our website. And we do have quite a few coming on the topic of AI in the coming weeks. So stay tuned. And then with this, again, here for you the contact details of our speakers for this webinar series. I think we have reached now the end of the very end of our marathon. Before we all leave, I just wanted to give you a quick heads up. As you close the Zoom, you will be taken to a short survey, which is anonymous, just a satisfaction survey on how you like the event. It will take less than 2 minutes, 5 questions, and we would really appreciate your participation. So this is it from my side.
Generally, also from my side, firstly, thank you for everybody. Thank you, David. Thank you, Philip, for joining in on and actually, providing the content for this. We couldn't have done that without you guys. And I think it's really, really interesting. I learned something, so that's really, really good. And yeah, for the listeners, please give us feedback in the survey, but also feel free to reach out to us. We're always happy to, hear feedback and improve what it is that we're doing, improve the content. Let us know if there's content that you would be interested in that we should try to prepare for you. We're always up for suggestions on this side as well. With that,
absolutely.
Thank you very much.
Thank you.
Thank you.
Bye-bye, everyone.
Thanks, everyone. Bye-bye.
Bye.
Bye-bye. Have a nice day.
Have a nice day.